Welcome to the slideshow on biotechnology. The chapter includes objectives, methods, and applications of biotechnology together with basic description of gene, its structure, replication, and protein synthesis. Biotechnology deals with the application of technology in big-scale production of biologically active products such as hormones, proteins, antibiotics, vaccines, medicines, etc. Genetic engineering is the manipulation of genes using tools such as restriction endonucleases, gene carriers or vectors such as plasmids, viruses, etc., and enzyme ligase. Genetic engineering is the manipulation of genes using tools such as restriction endonucleases, gene carriers or vectors such as plasmids, viruses, etc., and enzyme ligase. From the cell, DNA can be separated only after lysing the cell wall and cell membrane. The cell membrane is made up of cellulose, chitin, peptidoglycan, proteins, lipids, etc. in different organisms, and these can be separated or lysed by using suitable enzymes. Cellulase, chitinase, lysozyme, proteases, and lipases can be used for this. In plant cells, cellulase enzyme is used to lyse the cell wall made up of cellulose. In fungus, enzyme chitinase is used as the cell membrane is made up of chitin. In bacteria, lysozyme is the enzyme used. In plant cells, after lysing the cell wall and associated molecules such as RNA, histones, etc., using RNAs and proteases, DNA can be separated from the solution by adding chilled ethanol. DNA fibers can be taken out by spooling. The desired gene is cut out by specific enzymes, mainly the restriction endonucleases. Restriction endonuclease cuts the gene at the specific place. The isolated gene can be joined to another genetic material to form recombinant DNA. The desired gene is cut out by specific enzymes, mainly the restriction endonucleases. Restriction endonuclease cuts the gene at the specific place. The isolated gene can be joined to another genetic material to form recombinant DNA. The desired gene is
Gene cloning. Gene cloning by polymerase chain reaction or PCR. Steps involved in gene cloning are 1. Denaturation of DNA to single strands by heating. 2. Attaching primers or primer annealing using primers and enzyme primase. 3. Extension of strand by polymerization using Tuck polymerase enzyme. Tag polymerase is DNA polymerase from Thermus aquaticus bacteria. The steps are repeated many times to get adequate number of gene copies. Gene cloning. Gene cloning by polymerase chain reaction or PCR. The picture shows different steps in PCR to get numerous copies of selected gene. The picture shows different steps in PCR to get numerous copies of selected gene. Formation of recombinant DNA. Gene is inserted or transferred to another organism by using vectors like bacteriophage or through plasmids. The type plasmid or the tumor-inducing plasmid is often used for this purpose. This plasmid is obtained from Agrobacterium tumefaciens. Bioreactors. The recombinant varieties or genetically modified microorganisms are cultured in bioreactors to obtain their products in large scale. Bioreactors are large containers or vessels with arrangements for controlling temperature, pH control, etc., in which microorganisms can be cultured and their products can be isolated periodically. Needed nutrients and oxygen are also provided for the culture. Simple stirred tank bioreactor and spurged stirred tank bioreactor are commonly used for industrial purposes. Bioreactor. Some commonly used bioreactors are given in picture.
The session closes here and will be continued later.